Okay, so let's carry on the second part of this recorded lecture. Now, the steps of micropropagation. Again, let me clarify one point again. Micropropagation is a type of plant tissue culture. So the plant tissue culture is when you have a part of a plant, which we call an explant, and you culture it in vitro under controlled environment, providing it all the macronutrients and the growth hormones and the carbon source. And then you induce it to divide and divide and divide and divide. Eventually, this, this plant material, after a number of cell division, will be able to differentiate into a whole new plant of the same species from where it is coming. For example, if it's Dilbergia sisu, when you culture it, eventually it will differentiate into Dilbergia sisu, shisha plant. You know, for example, you have potato plants. Potato is one of the classic example of plant tissue culture and micropropagation. Pineapple. Pineapple is also one of the important plant. And these transgenic plants, for example, one of the best example of transgenic plants is containing plants, BT crops. The BT is coming from Bacillus thuringiensis, from where the toxin gene has been cloned into the plants through a TI plasmid vector. So micropropagation is a type of plant tissue culture because there are other types of plant tissue cultures. Selection of the mother plant, it is very important, as we discussed. It has to be disease-free, and you have to go for healthy tissue. You have to ensure that you are cutting an explant from a part which is clean. And then you will have the surface sterilization, treating it with a number of surface sterilizing agents. For example, alcohol, which is a very power desiccant, a dehydrating agent, would remove the water from the bacterial and the fungi, and it will kill them. Similarly, you can use bleach, you can use hypochlorite, you can use hydrogen peroxide, and there are also some other compounds that are named by the companies through different generic names. I do not know the exact chemical nature, but what they will do, they will not damage the plant tissues, and they will just wipe out, clean the plant material surfaces. But we have to ensure that there is nothing inside the plant as well. So this is why we have to pick a plant that is disease-free, it is healthy. You do not go for tissues that are really old, and you do not go for tissues that are really young. So this is a tricky part. Normally we take the apical meristems because they are disease-free, are very less viruses and bacteria inside them. Multiplication. Multiplication step is also very important. So when you have the explant cut from the parent plant, for example, here you can see the explant is cut. And then multiplication means when you culture this in a test tube or in a Petri plate, either in suspended culture or in a semi-solid agar medium, the cell will divide and divide and divide, and they will make an arrangement of loosely arranged cells. It would look like a number of cells that are loosely arranged into each other. That's called as callus. After callus, now each single cell of the callus can give rise to a new plant. How do you separate? One of the very common method is take a sterile blender and get them into a blender and blend them, and all the cells will be separate. And then, if possible, just pick cell by cell, or it's not so easy to pick cell by cell, but a clump of cells, a few cells, then a few cells, then a few cells, and then a few cells, and then you multiply them, and then you reculture them. For example, this is, you can see, this is the, let me take point. So this is the explant, stage 1. You have transferred the explants from the plant in vitro, giving them the growth medium. And you could see here, small shoots are appearing, callus culture, and from here you have to jump to stage 2, the multiplication. Here, it's very important that these singular plants, one by one plants, they are picked and uh, they are cultured separately, or at least in one while from a distance. There must be uh, ample amount of distance between them. Third part is rooting and acclimatization. This is also known as hardening. This stage is also known as hardening. This is very important as well because from stage 2, rooting and shooting and acclimatization, here you would give the plants auxins and gibberellate cytokinins to produce roots and shoots. Some plants we will give an ample amount, a mixed balanced amount of auxins and cytokinins, the plant hormones, and they will develop shoots and roots at the same time. Sometimes, for some plants, we are more interested to have rather more root formation than shoot formation. There we will have more cytokinin and less auxin, and the other way around. But from here, the plant has to be transferred to small test tubes because rooting is very important. These plants are really fragile. They have to be kept in these test tubes. For example, here you can see they are stage 3 rooting and they're still inside. They're still inside the in vitro or ex vitro, both are possible, but they still wait a lot of time in test tube suspended cultures. For example, here you could see in this one, this shoot formation is in the B and rooting C to E. So C, D, and E. And you could see that now an individual or a number of plants, not a huge number, they are separated and you could see these white roots. This is a suspended culture. This is a liquid culture and the roots developing here, and you can see a very strong root has been developed here. And how do we do this? You could see low auxin to cytokinin, and which means you have low auxin and more cytokinin, it will encourage shoot formation. But if you want to have a high root, you need to have high auxin and less cytokinin. And the last stage is the transfer to the plant new soil. You still will have to keep it in greenhouses, and you will have to be very careful because when we culture the plants in vitro, there's a lot of difference to what they are in the greenhouses or to the fields. For example, one of the basic differences is the humidity. With high humidity, the plants are not prone to to suffer any heat shock, and the stromata inside the leaves are not fully developed. So once we transfer these plants to the greenhouses in small pots, you have to be very careful because the cuticle, it will start getting developed and the stromata in the leaves will start getting developed so that they can start the process of transpiration. Because in the test tubes, they cannot develop these structures. So there are still a number of important days for them to stay in the pots before they can go and be a saleable or culturable plant in the field. It's like that. You can see it's A, B, C, D, E, F, G, H. And until here we discussed, and this is the part which is in the nursery. And eventually you can transfer them to fields. Composition of the medium is very important. And there are macronutrients, there are micronutrients. This is the carbon sources, of course, for energy derivation, vitamins, myo-inositol. These are some inorganic and organic supplements you really always need. Solidifying agents and the growth regulators. The growth regulators are always needed. Solidifying agents, there's only one solidifying agent used all around the world. There could be more, but we use only agar. And this is only ingredient that is not always needed. Growth regulators could also be avoided in case of some plants. You just have simple culture and you don't need in the beginning. These are some of the macronutrients that you need. Carbon you need because carbon is the backbone of all the organic structure. Nitrogen you need for amino acids, for example. Potassium you need for a number of enzymes. Hydrogen, calcium, phosphorus, oxygen, magnesium, and sulfur. Then the micronutrients that you need as cofactors for a number of proteins, enzymes, iron, sodium, chlorine, manganese, zinc, boron, copper, molybdenum, nickel, etc., etc. Energy sources, sucrose you can use, glucose you can use, fructose you can use, but mostly we use sucrose. It's a carbon source. And then the solidifying agent, if you want to work in a medium, semi-solid medium, or roughly commonly referred to as agar medium, we use not agar medium, solid medium, agar. So agar is actually also coming. It's a sugar. It's a sugar carbohydrate, but the plant cannot hydrolyze it, so they cannot derive any energy from it. It was just kind of a telling agent, and all the nutrients, the macronutrients and the micronutrients, they can diffuse through agar, and the plants can harvest them for their nourishment. Similarly, we use agar and semi-solid cultures in microbiology when we culture bacteria and some fungi like Candida or yeast or Aspergillus on agar medium. The growth regulators, very important: auxins, cytokinins, gibberellins, abscisic acid, ethylene, and some other, depending from culture to culture and from type of tissue culture, type of micropropagation, type of plant, and the type of development we need in the cultures. These are all used. But the two very important and always used are auxins and cytokinins. Gibberellins, abscisic acid, ethylene, and other plant growth hormones are also used. This is what I had already discussed about auxin and cytokinin ratio. In some cases, if you have intermediate balance, a mix of almost equal amounts, it will promote callus culture. Once we have a callus culture achieved, depending on the type of goals we have, if you want the callus to go towards shoot formation, we will reduce the amount of auxin and increase the amount of cytokinin. But at the same time, if we have this culture, callus culture, and we want to encourage root formation rather than shoot formation, we would provide them with high amounts of auxin and less amount of cytokinins.